Hi, Oliver. How are you?
Good, and you?
Good, thanks. Yeah, thanks a lot for joining. So we're just about to get started. Um, and okay, so it's 4 p.m. East Coast time here in the U.S. And I'm here with Oliver Bracko, and we're here to observe World Alzheimer's Day. So I have to be honest, I don't know the history of why World Alzheimer's Day occurs on this day. Maybe someone else on the call can or in the webinar can enlighten us. But it does occur to me that today is the autumnal equinox in the northern hemisphere. Um, and in terms of the four seasons, it's kind of it's the halfway point, right? So birth in spring, and then summer, and then we hit the equinox, and now it's kind of like middle age for the year. And for people who have the genetic form of Alzheimer's disease, I think middle age is approximately around the time, for at least some of them, when they might start to have symptoms of Alzheimer's. But as we know, Alzheimer's does not just happen in the genetic form; it happens spontaneously. And in fact, in most cases, it happens spontaneously, which means it's a disease that can affect anyone, and that's why it matters to everyone. So I am really grateful to have Oliver Bracko here. I've known him for a few years now, ever since I started working with Chris Schaefer and Nozomi Nishimura at the Cornell lab. And what I've discovered increasingly through this time is that Oliver is really the driver of most of the Alzheimer's research that's going on there. And I know there are some new folks in the lab who are doing that work, but I just thought we would take this opportunity while we have you with us. And by the way, thanks. I realize you came out of a lab meeting. You're sort of playing hooky from the lab meeting, I guess. That hopefully that's sanctioned, and we're really grateful that you could be here to speak about the research. So let me just start by asking you, Oliver, how long have you been working at the Schaefer Nishimura lab?
Yeah, it's actually, hey, thank you for inviting me to the event. I'm really happy to be here. Yeah, I think it has been now 5 years, in fact, and it's quite a long time. When I was coming directly from Zurich, from Switzerland, and had still the big idea in my head to working on stroke and using our microscopes to working on stroke and learning in memory, but somehow I got totally into the Alzheimer project at that time.
So you were originally studying strokes.
Actually, I had a fellowship to work on stroke, and if you want to stretch it, on post-stroke dementia, and wanted to know what's happening. If you early on have a stroke in life, you have a high probability to get dementia later in life, including Alzheimer's disease. And that was my idea, to use CRIS tools with my idea. But somehow it got driven into the blood flow deficits and Alzheimer's disease. And this is what I have done almost exclusively the last 5 years.
Wow, wow. It's interesting. So it raises all kinds of questions for me, because as you know, I'm not a biomedical researcher, and I'm very interested to learn more about the precursors for Alzheimer's disease. So I mean, could you just say in a nutshell, since this is where you started from, have you discovered any connections? Do we have any understanding today, either from others or your own work, about why people who have had strokes are more likely to suffer from Alzheimer's dementia later in life?
In fact, we don't know what is the connection to stroke and Alzheimer's disease. But what it is it's quite significant. It's at least, depending on the study and the clinical trial, can be up to 50 to 70% of all patients who had a stroke early on. So it's usually in mid-age. So mid-age seems to be a time where a lot of things are important that later leads to dementia or Alzheimer's disease. And the same is true also for stroke. So many studies have been done, including where the stroke occurs, in which regions the stroke occurs, but so far there just hasn't been a correlation really to you would think maybe a region of learning and memory, and then later on in life, that is the patient who gets dementia or Alzheimer's disease, but this is not the case, or at least we haven't seen it. I would put it that way. It
Hmm.
might be the case, but we just don't have the tools currently to maybe analyze it. And mechanistically, there's really not much known. So my idea was always what I was working on with stroke is that it's really interesting that animals that have a stroke, so they get first the physical therapy in a way, and you can do that also with animals. So they get a stroke, they have problems walking, they have problems with their eye and the tasting, and all those phenotypes you can get away, but what remains is the cognitive decline, and that's faster in animals than it is in animals without a stroke.
Hmm.
And my idea was to later look whether the vasculature or blood flow or any other phenotype then would remain the reason for why this is the case. But again, I haven't done it, so cannot say it. But what I can say is there's not any real model to do that at the moment.
Mm-hmm. I guess one of the questions I think that comes up in almost any field of scientific research where you're generating hypotheses and trying to figure out cause and effect is that two things that happen together that are correlated, um, that correlation, it can be coincidental, right? It can be just a random correlation that happens to show up, and so we ascribe meaning to it when there is no meaning. But if it happens consistently enough, then we start to ask, is one of those two things causing the other thing? Is the stroke causing the dementia? Does the dementia cause the stroke? But then there's always that third possibility, right? Which is that something else is causing both strokes and dementia. Something, there could be something, and maybe that's what you're referring to when you say, the phenotype, that there's something in a particular person in the way they developed, maybe in their genes or something about their environment that cause them to develop something in their biology that makes them more susceptible both to strokes and to dementia. And I take it from the kind of work you do that it seems to be possibly related to the vasculature. Is that about right?
I mean, I would say it's almost right. I wouldn't conclude with the ends that would probably say it's a vasculature, but it's not only the vasculature. I think that's an important lesson we have to learn, and we all know it, but we sometimes ignore it. That it's an interplay of all these phenotypes who probably drive the disease in that case. Let's say Alzheimer, or let it be the stroke. The stroke is also really complicated, but they are similar in a way that we really don't know that much about the cause. So we have a
Yeah.
lot of correlations in patients, and we have mechanisms in animals, but somehow they're not translatable so far. And I think the reason for that are probably many reasons, but one of them that obviously models are also not humans, but maybe many other reasons. If I would know that, I would do the experiment.
Right, right. And there are only so many hours in a day and only so many experiments you can run, right? So you have to be selective, and that's one of the reasons I think. So for me today, as World Alzheimer's Day, it's a day of gratitude to all the stall catchers volunteers who are playing stall catchers, um, and the fact that by accelerating the analysis of these data, that we, and by we I mean you and Chris and Nozomi in the lab, have the ability now to ask and potentially answer many more research questions than you otherwise could have in the same amount of time. It means you can, if you start to see something interesting, you can go a little further down that path without having to do that at the sacrifice of some other important research question you're asking. So maybe that's sort of the next question is, so what research questions has stall catchers allowed you to answer so far?
Yeah, I
Um,
mean,
I
that's
realize
definitely,
there are a lot. So maybe I
yeah.
can put up the slide that I think I mentioned to you. Let me see if I can share my screen.
If not, I
Um,
would be happy to share one slide before that slide. And I
oh,
think that,
yeah,
that
please do. So here, let me see. I'm going to see if I can give you the option to share here. So see if you're able to share that slide. That would be great.
I'm sure we will be able to share the slide.
Yep, I can see it.
Yeah, and the reason is why I created this slide is for another presentation, but I think it also gives the same idea here. It's not even talking about the details. It gives you an idea basically into my head what I think where the blood flow reduction and with that the capillary stalling stands, and what you can, I guess you can see my cursor for sure. So
Yep.
we have the brain blood flow. I centered it. It's not about saying this is the center of the disease and Alzheimer's is to the right. It's only for me, like I'm centering the blood flow reduction because this is what I'm working on. And you would have the disease and somehow they're interacting. And you can see now basically all the projects we are doing or planning or coming up with, and there are several reasons for them. So we have all this block here on the left, which is mostly the cardiovascular risk factors. And I mean, in fact, the stall catchers has helped us to work on 2 of these studies, and I'm really thankful for the players for really make it possible, and that's obesity and exercise. However, what is happening, the result was basically that it seems to be that the capillary stalling is not the link between these 2 diseases. First of all, the blood reduction itself, and also the Alzheimer's disease, but we have to separate. So what I'm trying to say is the idea is really to understand what drives it or what can drive it. It can be more than one. It could be a lot of things. And this is here on this side. And then we have the center, which is the mechanism. And I think the mechanism is that part we really need to understand if we think about any further, let's say, I don't carefully say the word of drug development, but it's kind of the idea of getting a target. And I think to do that, we really would need to understand how the mechanisms work on capillary stalling, on this blood flow reduction, and then on Alzheimer's. And that on Alzheimer's is extremely broad. So that means there are several mechanisms known to contribute to blood flow reductions. And I think we need to understand what capillary stalling is doing to those. And if we know that, we can think about what is the whole picture together with other Alzheimer phenotypes like amyloid beta plaques. And if we kind of know that, then I would say we can move forward in finding a target that really improves the blood flow.
So can I'm going to be the one to ask the stupid question here.
Yeah, go ahead.
That's my job. So the question is, so there are research questions, right? And you're talking about this kind of center area of the diagram where you're saying, there's some way that these things interact together in order to explain how the stalling phenomenon occurs, right? And then, so part of the question is how does that stalling occur? What are the molecular mechanisms that give rise to that? And then what are the implications for Alzheimer's dementia in particular from that? So in the course of doing these studies and answering these research questions, if you use a compound, a chemical compound, something that could potentially be a drug, and you suddenly see this reduction in stalls, and you suddenly see amelioration of cognitive symptoms, do you at some point say, even if we don't understand all the molecular mechanisms that can explain why that's working, if it looks like it's working, do we keep doing our research to better understand that, but do we take that outcome and say, maybe we should run a human trials on this, because why are we waiting?
Yeah, I mean, I think it's good that it's me one time here. I think if Chris would be here or Nozomi, they would definitely 100% agree with you. I'm also not at all disagreeing, but what I'm seeing is that this field in particular, but also other fields, this is the way it has been done over the last 20 years. And you can say there's nothing wrong with that, and I even would agree. So things that has been worked in mice has been taken into the clinic, and then the trials went through, and so far the trials failed. But that does not at all mean if we would move with our forward, it could be different. Of course, it could be really different. But I would argue against that only the same what I was trying to say, if we would understand the interplay of all these different phenotypes or hallmarks of the disease, I would predict that we could make a smarter decision for a target that we could choose. And I think then the probability is higher for this 10 years long process until you really get your result, whether it's working or not, and just would be with a higher probability. And I think
I
that's
see.
worth the time losing. But I'm at the same time not against this if somebody would like to do that, and I'm into it as well because I share the same hope. I also have the hope that it could work.
Yeah, I mean, that makes a lot of sense. So I guess, if so sorry, I'm momentarily distracted. It sounds like there are other folks here who might be interested in becoming part of the conversation. So what I'm going to do is we've got,
I can stop
we've,
my sharing.
oh, okay, okay,
Yeah,
thanks. Yeah, I'm going to go ahead and see if I can, I'm going to promote Agla, my colleague, to become the host. That way she can manage, bringing other folks into the panel so we can have a broader conversation. So Agla is now the host. That means she can kick me off. I better be nice. Um, so, so I think I understand that, there's potentially a huge investment involved in taking a potential drug and running it through this 10-year process. At the same time, I understand from things that I think Chris and Nozomi have said that sometimes the drugs that you're testing on the mice are drugs that are already approved for human use for something other than Alzheimer's disease, like chemotherapeutic agents. And in those cases, the pathway to getting those approved for a different use might be less. So I'm just wondering, and it sounds like maybe you've said this already, if there's a rationale for taking parallel approaches, um, um, for taking something that looks promising and maybe isn't the quintessential drug, maybe it's not reflective of a complete understanding of the underlying molecular mechanisms, but if it can make an impact, in 3 years instead of 10 years, while we continue to do that other research, um, is there a rationale for pursuing that? And
yeah, I think there can be a rationale for doing that. There have been examples that have been done not in Alzheimer's disease because there we don't have a new drug since a long time, but in other examples exist. I mean, again, I'm definitely not against this. Um, I mean, I'm a little bit also not against this, but I think it's really like a broad drug that has so many targets that we think it probably can act in, I make it up, 5 different diseases, then I'm already skeptical. But there are some examples where that in fact really worked. So I think so, yes, if somebody can push it and one would move it forward, I'm definitely not against this. Um,
yeah, yeah,
but I've seen a lot of frustration. I mean, I know a lot of folks in the field that have their amyloid beta antibodies, and I know their publication 10 years ago was a lot of investment. There was nothing wrong, and that's even good examples. I want to bring up the examples, not like they didn't work. The antibody comes in, it takes out the amyloid beta plaques of the brain, but there's no improvement, at least with that cohort of patients that have been tested. There's now one cohort where that might look a little bit different, but that needs to be further analyzed. So I think there
yeah.
are definitely reasons to maybe push it earlier.
Got it. Yeah, that makes sense. And I think that's a really important point that you just made, which is that you could have something that in an experiment, reduces a hallmark characteristic of Alzheimer's disease that reduces plaque formation, for example. And then you think, oh, well, it's that correlation causation thing. You think, well, if you reduce the plaques, then you're reducing the disease, but it has no impact on the symptoms ultimately when people start to
But I
use
want
it.
to, just say that this has been shown many times. But I want also to point out that in mice, it really reduces all the symptoms if you take
Mm,
out those amyloid beta plaques, and also in some other animal models. So in animal models, that was extremely promising. It just then didn't turn out to work in humans for many reasons that I also don't know. And we cannot look in most of this data of this current trials where we maybe get some more idea, but I'm sure they get republished with time. But I'm just saying those antibodies have worked in, or drugs have been working in mice, and they have been cognitive improvement, reducing amyloid beta plaques, reducing inflammation. So these drugs work in animals, but so far just it hasn't worked in human patients, unfortunately.
Yep, I think that's a great reminder. All this work and all these promising findings are in mouse models of the disease, and we don't really know yet how they might work in humans. So it sounds like I'm just now seeing some notes here that others are raising their hands to speak and might have some questions for you. So Agla, I'm hoping you can bring someone into the fore, or if you already have the ability to turn on your mic. Looks like maybe Guy, did you have a question or a comment? This is Guy Calkins.
Not at this time.
Oh,
No, I
okay.
think we were just getting ourselves hooked up here. She was inviting us in and we turned on, so I'm good. Thanks.
Okay, gotcha. All right, thanks for letting me. And by the way, it's really great to. I saw. I guess Guy got the email about this hangout, and he dropped off about 6 months ago because of technical issues in playing stall catchers. And he saw the email that Agla sent out about this hangout, and then came back on, and it looks like it's working for him again. So it's really fun to see Guy, back here with the community. We missed you, and you helped us out so much in some of our earlier stages of the project, not just in playing stall catchers, but in helping us get great engagement and participation in the megathon. So we're super grateful to that. So just to kind of, I'm going to ask the hard questions because I'm thinking, maybe I'm the only one who has these questions, but I'm guessing that a lot of members of our community who volunteer their time want to know this. And when we began the project, I had this, maybe it was a naive idea that if you've got these questions that you need to answer, and we can really speed up the analysis, in a few years we might have a treatment target. So it's been a few years, and I believe that we're much closer than we were before. So I'm going to show you why I believe that. I'm going to show you this other slide, if I can share my screen here, and then I'm hoping you can kind of either temper my enthusiasm or bring some realism to what I'm saying and to all of us here. So I just have to move this thing out of the way so I can show the slide. All right, can everyone see this slide? Can you see it, Oliver? Yeah. Okay, so this slide, Chris showed me this slide, and I got very excited because, this is one hypothetical, molecular model of the molecular mechanisms, these things, that the causes and the interconnected things that all lead to the stalling process, right? So this is an idea. I think this diagram represents a very educated guess about how these stalls are forming. And then where you see these green check marks is where we use stall catchers to gather data, to analyze data, rather. You gathered the data, Oliver. We analyzed the data, or we started to, and then you completed that analysis. And then in the green check mark means, yes, this part of the guess was actually correct. We're right about this part, we're right about this part, we're right about this part. And this was as of about 4 months ago. And then these red question marks represent some outstanding questions. And once we answer these 3 other question marks, then I think we have a relatively complete picture. If those end up being green check marks, meaning, yes, those were also correct, then, so I guess the first question is, how close are we to answering those other questions? Do we already have data sets, or do we need to collect data from those?
Yeah, I mean, those slide is first, of course, the simplification, which is fine, and the connections. Some of them has been shown in other systems or in other organs, and some of them has been shown in the brain. So I just want to point out that this is, of course, what we think at a given time. But to really show that this is happening, we need the partners in between. And we try this with several ways. So we have done several experiments since then, and we don't know the results, but they haven't been creating data for stall catcher. And one reason for this is that I think this is an excellent map. And since we know that some of these genes like VGF or NOX2 is decreasing the number of capillary stalls while improving blood flow, we still looking like, like into the universe. You pick a star and then we look at it and we see, is it somehow involved? Yes or no. So the idea is to look into all of the stars. so in all of the genes or all of the proteins in those cells, and see whether which are the ones are changing from a stall to a flowing capillary. And why I'm thinking this is a better moment is maybe we are looking into this and this has good promise, but if we would look from outside looking into it, we will maybe missing the big players just because we cannot fish them out with our current approach. So what we're doing is we try to look into all proteins at the same time, all genes, and then compare stall to flowing capillaries. And I think what I hope, of course, and what I wish, that exactly those turn out to be the ones who are changed. I mean, those, and I predict that this is going to be the case, and probably it is, but there might be others. And the interplay, in particular, if you go down your chart, if let's say from a NOx, from a VGF, and you have this eNOS with a question mark, eNOS is one player who could be there, but there could be 10 different pathways who could be downstream of this and could lead to this effect further down. And this is true for each of those steps. And I think if we try each of them error and try, I feel like this is a good way and it's possible. But there might be other ways to also approach this way. And the technology is there for doing that. And we have a collaboration with another lab who is specialized in getting those blood vessels out of the brain. And this allows us to do such analysis.
Yeah.
But yes.
Yeah. Okay, so I just want to make sure I understand, because you've said a lot of things, and I have a very simplistic view of this, and I'm trying to expand that view now. So and hope that I can bring others into this with me. So it sounds like what you're saying is that you know there's other research going on, outside of mouse brains, maybe other organs in other mice or other animals, that helps to feed the overall understanding of what's going on. That there's a lot of evidence coming from different places that helps us get a better understanding of how this works. And I guess one thing that isn't quite clear to me is, so for these remaining question marks, are these intended to be brought into stall catchers, or do you expect these answers to come from other research that's going on?
So sooner or later, these, or let's say the question mark, but it has a different gene name, definitely goes into stall catcher for sure, because that's always our final answer is, does it improve blood flow by reducing the number of capillary stalls? And the way is just like the question marks are here now, and there are some names like eNOS, but for real, it could be definitely another one or another one. And I think
I see.
that doesn't change the fact that once we have an idea, what is this question mark? And I think this is an educated guess, because we have some indication in particular for this eNOS that is involved. However, the names might change to simplify it in this way, but it's definitely will get data sets. And I think this should be relatively soon. We also have there's no shortage on data sets, but those are for me like also the interesting ones, because I also want to know what is going on in a stalled cell, or in a cell that just installed.
Yeah. So okay. Well, thanks. That's very helpful. I'm gonna stop sharing this diagram and my screen so we can go back to the normal view. And so it sounds like you're saying that that we will see something like that. We might not call it eNOS when it shows up in stall catchers, but it one way or another, it'll end up in stall catchers, that part of the model will get tested in stall catchers. And that there could be other things like eNOS that have a similar impact on the overall model that could be investigated. I think it brings me back to this question of, when is good enough good enough? So I'm an impatient optimist, right? And I've stole that from Bill Gates. I heard him talk a few months ago, and he self-referred as an impatient optimist, and I love that because I feel like I really relate to that feeling, and I think anyone who's struggling with disease in general, chronic disease, and particularly Alzheimer's disease, there's this sense of frustration, right? We don't really have an effective treatment. We have treatments. I don't know, how effective they are today, but we want something that's going to really make a difference. And so, you know, so I think on the one hand, I really appreciate kind of as a scientist and as a pragmatist, this idea that, well, before we really invest in this whole clinical trials process and human trials and getting something approved, we should really we should feel very confident that this thing is going to work, right? That we've sort of evaluated it from all these different angles. But then there's a part of me that continues to feel like, if we've got something that looks promising, even if it's a 50-50 shot, getting it into the pipeline sooner rather than later, could impact a lot of lives. And I realize that, there isn't probably nobody has the perfect answer to that question or that dilemma. It's really balancing resources so, okay, let me distill all of that down into this question. How close do you think we are, based on the work that you're doing today, to having a treatment target that we could bring into clinical trials, like measured in months or years?
Then it's definitely years.
Okay.
I would give it definitely years. That I give you one reason why I think it's definitely one thing which we definitely lacking is do capillary stalls occur in human patients? And of course, that's a step that we don't know, and it's not easy to answer at a given time. To do that, it requires imaging technology that kind of exists. So I'm optimistic there that we can soon start figuring this out. So and I definitely believe that it's going to be the case. Whether it has the same impact or the same role as it is in mice, that's maybe we cannot figure that out ever before we would start like trying to do a clinical trial. So I'm on your side. I say years because I don't want to say anything else, because it's going to be years even if we start now. So I definitely I would give it a years, but I don't want to give a number because I also don't know. That would depend on results. I just would be careful. So it's definitely years. So there's no way around this. I have to say that. And I also see the dilemma, and I'm not totally not against this. I think you can try both, and you should try both. I mean, science can always be a educated lucky shot, but it can be also not. And there might be, I mean, I don't want to give too many negative examples. I really want to stay positive. So I think there are certain ways to make things faster. But it requires an understanding that's a little bit deeper as what we have now.
Yep. No,
Yeah.
that seems like a very fair and reasoned answer. And it looks like we have some questions lining up. I had another question for you, but I'm going to hold it. And why don't we go to, so Guy Calkins has a few questions here. I'm going to ask them both, and then we could take them one at a time. So his first question is, how are we distinguishing between causation and correlation? Do we actually know that lack of blood flow causes Alzheimer's, or is it just correlated? It doesn't sound like we're actually sure that eliminating stalls will actually affect Alzheimer's. So maybe start with this question from the standpoint of the mouse models of the disease and the work that you've done with mice.
Yeah, I think that's I think that's an extremely sharp question to ask. So it is a correlation it's indeed a correlation, and of course we try to work on proving that it's not only a correlation as the work we are currently doing on the mechanism of capillary stalling. As long as we manipulate it simply and we reduce the number by seeing that blood flow improves, it is a correlation. And the same is true for the cognitive improvements we are seeing. It's also a correlation. So there are two levels of correlation. So in some of things we can exclude better than others. So the flow correlation to capillary stalls, I would say, is a strong one. It's still a correlation because we don't really know what is at the site of stalling going on. And this is a technical problem that we hardly can overcome at the moment, to really get that cell, these two cells or more than two cells in a given time, and where this is occurring. when we really can grasp those cells, I think we can answer this question to correlate the stalls to the flow. The correlation and then to the cognitive improvement is that's a really tough one, because I mean, you must imagine you have a blood cell with blood, and then you have all the different brain cells in this area, and they are affected. And if you have reduced blood flow, they have less glucose, so less sugar, so they can be less active, and we call it hypoxic, meaning in this area is less oxygen, and this affects those cells. And there's some way to the neurons and to the cognitive function that we also don't know. We have a project working on that, but so far that's a correlation. And how does it really correlate to Alzheimer? I would say that could be said positively and could be seen negatively. I would say the positive is that we see it in our Alzheimer mouse models, and the only, despite those are models, of course, but the only difference they have to any other mice is that they have a certain form of Alzheimer's disease. So there's a strong correlation. If you would say, is it only in Alzheimer's disease? I would argue it's not only probably in Alzheimer's disease. I think that the blood flow reduction is seen across many diseases, in particular all the neurodegenerative diseases, which I think is really interesting, that puts it into a player maybe that triggers diseases, this triggers cognitive decline. And there's this idea of a two-hit model where you would say, let's say you have blood flow reduction and you have amyloid beta plaques. If they come together, it makes the whole progression faster, which leads to Alzheimer's disease. Let's say you have another hit, and let's take Parkinson's disease, where you have deposits called by protein alpha-synuclein. Let's say you have that hit, and that triggers the direction into another disease. And I think that's really interesting, but it is a correlation. And but I would see that positive.
Yeah, right. And so it's interesting because I mean you can sort of look at the experimental results and from the standpoint of correlation and causation, and you can say, you know, when we have a mouse that, so we have two sets of mice, right? One set has Alzheimer's and the other doesn't. And we see that the set with Alzheimer's has memory issues and other cognitive issues compared to the wild type mice, right? And so, you know, based on the evidence, we believe the reason they have those symptoms is because they've been given transgenically the human version of Alzheimer's disease, right? And we see physical manifestations of that as well. So that appears causative, at least that part, right? Then we take those mice that have the cognitive symptoms and we give them a drug, and those cognitive symptoms disappear. That looks pretty strongly causative. You know, you give a drug, the symptoms go away, you know, as far as the evidence supports that. So the part that we're not sure about that could be correlative or causative is whether the reason those cognitive symptoms goes away is because the drug makes the stalls go away. We see that we count the stalls and stall catchers. We see the stalls go away, and we see that when the stalls go away, the cognitive symptoms go away. We just don't know if those cognitive symptoms are going away because the stalls are going away or because that drug is acting in some other way to improve cognition. Would you say that's a correct
yeah, that's
interpretation?
100% correct. And this is also what we cannot answer. And I would almost give that a bad luck that that's a protein which we started with, which is called Ly6G, which is a surface protein of a white blood cell. And even it's starting with this, that nobody really knows what this protein is doing. And that obviously makes it difficult. If you take, though I want to give a negative and a positive, the positive is that there's now some data that links this protein to a process called this blood-brain barrier. And the blood-brain barrier is the barrier between the brain and the blood vessels. And this is quite tight. And it's known that it's less tight in patients with Alzheimer's disease. And this is less tight and simplified. You can imagine things can go easier into the brain, things can go easier out of the brain. And if this protein would be involved in that, it could well be that it's opening up this blood-brain barrier or destroys it. It's not as simple as open and close. It's transporters which are there, and they're known to get reduced with aging, and they're known to get faster reduced with Alzheimer's disease. So some connections we can come up with, and in fact, this is also what we're really trying to do. And there are several other indications from patients and from recent clinical trials that links this blood-brain barrier to Alzheimer patients and blood flow reduction. So there's a lot of things going on, but I say the timing is, and each of them you need to test, of course, and that just takes time. But I think there's a picture emerging, and on the most pessimistic side, you could, or pessimistic and optimistic, if you feel, you could say, even if it then would not be the stalls, that would be okay. You just need to figure out what is else doing, and then you go for this. And you could say we lost a couple of years why this has been happening, which is true. But in the end, you find a way to explain it, then it's good again. And you
So,
may lost the time, and I understand that this is kind of not satisfying for anybody. I mean, we are a family with dementia, genetically as well. But it's nothing to change in a way. We could say we hire more people, do much more, but every scientist with this research project would say the same. And so really there's a limitation. And I'm not saying only the money, also the persons who can do it. So I think it's just the nature of it. So we can be wrong, but then out of this being wrong, we can probably learn something as well and move on. I call that science, and I think that's why it's also really interesting.
Right. Yeah, that makes total sense. You have to sort of place your bets on where you think, the research needs to happen, and you might go down a path and you get a negative result. And I think one of the things that's gradually becoming more appreciated now in the scientific community is the value of negative results, that they represent time and effort, and they represent, a finding that then helps us realize, okay, we've ruled out this one case, so now we can refocus in these other areas, and there's really no other way to do that, I think. And I guess to me, kind of, summarizing what you said is, if you end up discovering that, well, this drug is helping get rid of cognitive symptoms for some reason other than stalling, but it's still getting rid of cognitive symptoms. My take on that is, if you give me snake oil that makes my dementia go away, I don't care how the snake oil works. I'm just glad that it's making it go away. And I think that's the positive, right? If in the course of studying stalls, you find a treatment, whether it's related to stalls or not, you found a treatment. So I think that's huge.
And so far, that's kind of true for all the targets, but with different angles, which is interesting. You would say it's NOX2, it's expressed in many cells, has many functions, which are quite diverse, let's say, from this Ly6G on the white blood cells, or at least that is what we think. In fact, we don't really know that. Maybe we're just missing it. But then you could argue, if the effects are quite diverse across different targets we are choosing, that would speak for specificity for the stalling phenomenon. I'm not saying this is the case, but that at least would suggest it at the current stage. Yes.
Yeah, that's exciting. That's very interesting. We do have another question here from I think from Mike Caprero, whose name you probably recognize as one of our very prolific catchers, like Guy Calkins. So he says, so we are nearing the end of the current structural mapping data set. Do you know what the next data set will be? I don't know if you can answer that. Maybe you can, but I think Agla might know the answer to that one. And if not, I can possibly look it up quickly. We do have a data set in hand. So it's already been transferred from the lab, but we still have to pre-process the data set before it can go in to stall catchers. That's a machine-based process. Last time we did the pre-processing, it took an entire week and cost $1,000 worth of cloud computing. So there is a computational aspect to what we do, and it's not trivial. so I'm not sure. I'm waiting to see if Agla says anything, if she knows about the structural mapping data set. Maybe Agla, if you could check Mike Lamont's text messages on the other Slack, maybe we could find the answer there, unless you know what it is already. In the meantime, Oliver, there's another aspect to this. So I have a friend who has had some various health issues and might have a cognitive component to those issues. And she said that, I was talking to her. She's always asking. She said, I hope you're making progress. I need your answer. I want your answer soon. And I said, well, we're looking at different things. I said, it's interesting. It looks like some form of inflammation might be implicated. And when we've given mice certain drugs that address that inflammation, some of their cognitive symptoms go away. And I said, obviously we don't know if that would hold for humans, and we don't know exactly why. And she said, well, I'll tell you what, she said, I was taking, she said, I have some autoimmune issues, and I was taking a drug that would suppress my immune system a little because of these autoimmune issues. And she said, and when I did, she said, my cognitive symptoms went away. So now that's just one person, right? And that's subjective, but this is becoming kind of a thing now, right? This N of one, precision medicine, each person, is a unique organism. Everyone has their own genome, their own biome, and all these, and all what they call the multi-omics and proteomics and so on. And everyone has their own unique signature. And so, something that works, some treatment that works for one person might not work identically for another person, or even at all. But I wonder, are we really tapping this growing base of knowledge? Because each N of one by itself might not have the answer. But if you look at that data collectively, is there something we can learn about Alzheimer's disease and how to treat it from, the methods of precision medicine and maybe even some of the anecdotal data that's out there?
I mean, many scientists are doing this now. I mean, we have all these huge data sets, and first of all, it's not so easy to combine them because for technical reasons, but the people who are overcome those, I mean, there's a lot. I saw, for example, a study today coming out looking into MRI data. So looking for structural changes in brains of Alzheimer patients, and they looked into patients who have a family form, so early onset Alzheimer's disease, and patients who have sporadic Alzheimer's disease. And it was thought that there are quite some differences, but this study, for example, suggested if you carefully look, have a new measurement, take, let's say, 50 studies together and look into the brains, it looks extremely similar. Which is kind of feels logic, I understand, but at the same time, the thought wasn't that there's different areas affected at a given time. But if you have a huge patient pool, then it looks similar again. Now, I can argue it depends on which patient you probably look in, and that is true. The same argument is what the company is using for those drugs, which I also totally agree on. Maybe the patients that has been selected in those clinical trials couldn't be really carefully selected because we don't have the biomarkers to perfectly select Alzheimer patients. And there's a lot of definitions going on. And for a clinician, it's relatively clear what an Alzheimer patient is, but from a neuroscience standpoint, it's maybe not. And I think there's a lot of things that we learn from those trials in any ways, how to do a better one in the next round.
It sounds like precision medicine has a lot of challenges, not the least of which are data science challenges, and just dealing with these heterogeneous data sets and sources that are difficult to combine and to analyze in combination. But it'll be exciting to see, as we get better at doing those things, what we can learn from that. We have some follow-up questions here,
Yeah,
I think.
I'm just seeing.
Yeah, so Guy had another question, and then it looks like Martha's got some questions as well. So Guy was asking, he said, you stated that one particular treatment that eliminated stalls in mice did not have the same effect in humans. Did you mean it didn't eliminate stalls or didn't lead to improved cognitive function?
Okay, that's almost what I was trying to say. Maybe let me specify that. So wait, I read it again before I say it may be wrong. So these effects, all effects we are seeing are in fact effects in mice. So in mouse models of Alzheimer's disease, we don't have a human effect yet. And the reason is we just don't know whether humans have capillary stores. But what I can say for sure is that I would say almost all, but that may be also a matter of definition, since Alzheimer's disease is probably not one disease. It's sort of a syndrome, if you want, and have largely all of them have blood flow reductions.
So it's interesting because it Chris has always said, since the beginning of almost since the beginning of Alzheimer's disease, we understood that there's this 30% overall reduction in brain blood flow. And what you're saying is that, I think what you're saying, and correct me if I'm wrong, is that yes, we do see this reduction in brain blood flow. We just don't know if in humans it's being caused by stalled capillaries. We've only shown that that's happening in mice because we literally haven't looked in the human brain at individual capillaries and counted them the way we're doing it with the mice. Is that what you're saying?
The reason is we can't. And one reason is we just simply can't. And the technology doesn't allow us to do that at the moment. And the technologies that measure something like blood flow just don't have the resolution. They take large areas and measure, let's say, large tissue changes, perfusion, is it called, for an MRI, for example, or for PET imaging, where you measure the glucose and have a tracer. So we know they have a reduction, but we cannot grasp the capillary level. And it's of course not only, so this is a trend at the moment. Let's say we are looking for capillary stores. Other people have seen capillary stores, but of course there are other hypotheses that also suggest where the blood flow deficit is coming from. And I think that's extremely interesting because first of all, it's rarely one in biology. So it's usually several mechanisms working together. But I think what is interesting is this emerging field of looking at the smallest vessels, because first of all, that are the most vessels. This is really where the oxygen exchange is occurring. And what definitely is many data sets pointing towards is that these vessels are important for the blood flow reduction and also for the disease progression. And then I would come back to, it has a component of stalls, it has a component of constriction, and it has a component of different things, and it all comes together. But on a science perspective, it's just, I think it's emerging together, since we can do it now, at least in mice, but at least that's the starting point for now. We can see that those vessels are really important, and they haven't been implicated before. And one major reason is just simply we couldn't have done it. So other vessels have been implicated in blood flow reduction, and they're also involved. But it just turns out that the picture is much more complicated. It's bigger. And if you see the capillaries, it's mostly about 80% of all the vessels are capillary in the brain and in the whole body. Wow. So this is really where the exchange occurs. And now you can think about all these organs which are highly capillarized, like all the ones you think about. There might be similar mechanisms in play, like the eye, the lung, the kidney, all the ones where there's highly oxygen consumption. So I think this is really, really, really an interesting field and can spread quite far. So there's a lot of implications we can take from there.
It's sort of it's interesting that you mentioned the eyes, because I've heard it said that the eyes are the only part of the brain we can see, right? That parts of the eye are the early visual system. It is a part of our brain, right? And so is it conceivable that we could go to our eye doctor, they could look in and see the capillaries inside our retina and actually get a signature of capillary stalling in the human brain by looking at the eye?
Yeah, I mean, we have a grant that got rejected for funding for just giving that question to ask.
Oh, wow.
But that doesn't mean that's nothing rare, I would say. That's just how the process is. Maybe it wasn't that good at the time. And yes, two things. It can be, we can do it in mice, and several clinical institutions are trying to get the resolution on capillary level. And I try to avoid spreading more, not the ideas, which is fine, but spreading the idea of the eye is interesting, because the eye is one of, it's a window into the brain, that is true, but it's the only place where you have a stalling-like effect in patients. And these are patients with diabetes. And the patients with diabetes, they have white blood cells that adhere to the capillaries of the eye. And this is known since many, many years. But it hasn't been really followed up. But there's quite some data showing it, and from historic pictures, and now also from newer data. And you can really nicely see from some groups, from Rochester, for example, so they have an imaging technology where you can see slow or blocked capillaries in the eye without a labeling, because labeling is then for patients also a problem, because you cannot inject those tracers into the blood system. So there's a lot of those. So that's definitely something extremely interesting. And now I want to kick, but I don't want to spread too much optimism. But it's my furthest optimism I can see, because what patients get to treat the macular degeneration or the diabetic retinopathy is, in fact, they get an anti-VEGF antibody injection into the eye. And this is working extremely well. So these patients can see much better. It's extremely effective, and it's commonly used across the world now.
Okay, I'm going to put this on my calendar. This is the day that Oliver made an optimistic statement about no, that's a very, very compelling and interesting finding. So it sounds like what you're saying is that basically the same mechanism for stalling that we seem to observe in the mouse models, in the capillaries of the mice as we're playing stall catchers, that same kind of white blood cell adhering to a capillary is what happens in the retinas of people with diabetes.
Mm-hmm. A certain form of diabetes.
A certain form of diabetes. Yeah. And then when you put the VEGF, which is a familiar acronym because we've analyzed those data in stall catchers, when you put that in the eyes of these, I'm not going to ask how you do that, but when you administer this to a diabetic patient, that improves their vision, that it seems to alleviate that stalling.
That link is of course missing.
Okay.
Yeah.
Okay, that's the optimism part.
Yeah, that's the optimism.
That's the hopeful part.
I didn't go.
I want to make
sure.
Okay,
let's say it's this way. Let's say I can flip it a little bit. So you can see that the problem is the blood flow measurement is a little bit missing in those capillaries. As soon as, and I think lots of groups trying to get those measurements, and I think then we will know whether the mechanism could be the same. Of course, I would say it probably is, but it has been shown, and I cannot show it at that moment.
So Guy is responding, he says, so you are really saying it is oxygen deprivation, not necessarily blood flow reduction. And stalling is one possible cause, and hopefully a good candidate, of the oxygen deprivation. Would you say that's about right?
That's about right, but I want to just I
want to make sure we get to Martha's question, because we are almost out of time. So maybe if you can answer that part quickly.
Okay, I can answer that part quickly. You would have a blood vessel and a cell is blocking it, and the blood is carrying the oxygen. And if you block it here, so all this tissue follows this blockage, they cannot get the blood. So for that time, when it's not flowing, they don't get the oxygen. So that is what I'm trying to say. That could be true for all of the different genes we are looking at, because they all have the same effect, but the effect would be, and in fact, a collaborator of ours has data showing that when it's blocked, there's less oxygen. So that's really nice piece of data. Okay, quickly read Martha's.
Yeah, so Martha's, yeah. And so, Martha Taggart, she's the science communicator extraordinaire from Bright Focus Foundation. You may know Martha already, because of course Bright Focus has not only been a supporter and funder, original funder of stall catchers, but also went on, I think after learning more about your work through stall catchers, they went on to support some of the work that's going on directly in your lab. So we're absolutely grateful to Bright Focus, for the tremendous support over the years in making this project happen. And it's kind of overwhelming to be here, with folks from Bright Focus, from our catchers. It's the whole village thing where everyone pitches in different ways to make this happen. And it's just such a tremendous feeling of gratitude and humility to, kind of be in the midst of all these great people. So I'm just grateful to everyone who's been involved in this. So sorry, I'll go ahead and read Martha's. So she said, Oliver, what have you and others discovered about VEGF and Alzheimer's? Does it go up or down? Is it related to plaque formation? This is of interest to me and Bright Focus because of the VEGF role in macular degeneration, which is one of the 3 diseases, by the way, that is studied by Bright Focus, other than Alzheimer's and glaucoma. And she says, that is overgrowth of fragile, leaky blood vessels, creating a messy, inflammatory situation in the retina and ultimately nerve cell death. That might make more sense to you than it does to me. That's beyond my pay grade here.
Yeah, Martha knows what she's saying. Yeah, I mean, I definitely also hope this is exactly what I would say. This is what I would predict what is going on, and I also would like to continue that in the future. Thinking about this question, but this is exactly, and I think the way to approach that, and I think Bright Focus would be, of course, optimal for that, but would need some collaboration to do those experiments. And it's not so easy to image the retina, also in the mice in particular, and the same in patients, but it's exactly the mechanism. I would say it's a leaky. So the eye then also have, is what I said, this blood-brain barrier, and VGF is known to open the blood barrier. In fact, it's one of its function next to creating new blood vessels. And interestingly, in Alzheimer's disease, we're not getting new blood vessels, but you have more VGF. Now, the data in mice regarding VGF or anti-VGF treatment is quite diverse, and it really depends on, for one hand, it depends on where you inject it and how do you administer it. And has diverse roles. So if you inject, that gets really into the details, but it can improve cognition, and it can also reduce the number of plaques, to simplify it. But that's really simplified because it's a really, really complicated and a broad spectrum protein that has a lot of functions, unfortunately a little bit, but that also makes it really interesting.
It's just amazing to me, to hear you talk about this stuff. And thanks for answering Martha's question. Just the levels of detail and complexity underlying the human physiology of any organism. I'm just so glad there are people like you who can study and understand that stuff and do this great research. And honestly, I think we're wrapping up. We're a couple minutes after the hour. But I'm really, again, so thankful to you for, Oliver, for joining us for this and answering these questions. I've learned a lot. I hope others here have too. Some really great questions from folks. We've been doubling points all day on stall catchers. We used to just double them for 1 hour, but then that was never the perfect hour for anyone. And we have people all over the world playing. So we doubled up the points around the clock. I hope everyone's enjoying the double points and taking advantage of that. And I know some of the folks on here, including, Mike and Guy, have been known to continue to play stall catchers while they're on these hangouts, just because, you don't want to miss those point opportunities, and they're capable of that level of multitasking. So anyway, I think we'll wrap up here and say thanks again to everyone for joining, being part of this hangout. And we're recording this. We'll share it out so others in our community can access this information. Do you have any closing comments, Oliver?
Yeah, I do. And I think I'm extremely thankful for all the players playing the game. And I just want to share also my feelings. So you see the same how we are feeling. You see how optimistic and really beautiful it is if you see that the phenotype is reducing the number of stalls, blood flow is coming up, the data set is showing what you would like to see, we are all excited and it's moving forward, and that's the extremely positive part of being a scientist. But you also see, and I think that's extremely important, how frustrating it can be at the same time. You're counting, it doesn't show a result, it doesn't fulfill your hypothesis, and you need to adjust your hypothesis, and months of work are gone and you didn't get a result. But this is just a matter of fact. That is how we scientists live. It's an up and down, and I think you feel the same, and I can see it also in the comments, and I also understand it. But I think it's important for you and for us that you see this is how the progress goes.
Thanks. I appreciate that. I
Mm-hmm.
think that's both sobering and kind of hopeful and optimistic at the same time. You know, we expect that there will be some dead ends along the way, but overall we're making good progress. And so thanks again for sharing all this with us, and hope to have you back for a future hangout.
Yeah, sure.
So
I'm happy to join again.
great. All right. All right.
Thank you.
Hope
Bye.
everyone enjoys the rest of their days. Bye.